So thank you for the opportunity to talk about the work that was done in my lab. I am not a microfluidics person. I'm going to talk about macrofluidics today, so doing work that we did on the benchtop. So my name is on the presentation list, but really this whole project is driven by a very talented graduate student who was in Krish's lab, who is now defended his PhD earlier in April this year, Mohammed Ibrahim. And he was very, very brave in sort of getting this collaboration up and going. So we're going to talk about the work that we did modeling benchtop biochemical attacks and present a solution that we came up with and sort of show you a little bit about the feasibility work that we did it. But really the subtext for this talk is curiosity, communication, and collaboration. So as I said, Mohammed was really brave, and he really wanted to understand where the potential risks were in doing benchtop work and thinking about that in his projects for applying that to microfluidics. So his curiosity was key. Our communication was really crazy. We had very different definitions for words. And so it took a lot of patience in order for us to really get to the last C, which is collaboration, to show you where I am today. So don't be afraid. I learned a lot about electronics. I learned a lot about microfluidics and things that I never really imagined delving into. And I think if Mohammed were here, he would say he learned a lot about pipetting and, and putting on gloves and things like that. So I'm very excited, and we're going to continue on with this collaboration. So what are we going to talk about today? So the real driving question in our work was how trustworthy is analysis on a biochip? So we talked a little bit about how doing this all automated eliminates personal errors by the operator. But what else can we take that a step further forward or take that a further step back? And so what we started thinking about were biochemical level threats. I mean, what kind of countermeasures could we come up with? So what I'm going to talk about, I'm going to do this in two parts. I'm going to model some different particular threats and what that would look on the bench. And then, again, at the end, we'll talk a little bit about the counter-level defense ideas that we came up with. Okay, what are the risks and how can they be minimized? Okay, so really any experiment, any experimental protocol that's used in a clinical lab or in forensics is going to go through some very basic steps. There's sample collection, preparation, the amplifications, which is going to be done on the machinery, detection, and storage and disposal. So we heard a little bit about maybe some of these endpoints earlier this morning. What we were interested in focusing on were these early bits where things could be attacked. So where at the biochemical and bioassay level, what are those vulnerabilities and what can we do to prevent those? Okay, so our objectives were to model that, and then once we modeled those attacks, really try and figure out which is the most stealthy, and which is going to drive the second part, how do we stop them? All right, so I've learned to not assume that you know anything, so I'm going to go through some really basic steps of really how we isolate DNA and PCR, really what it is, and how we visualize these. So bear with me a little bit if you know it. So really, what are our basic steps? And first step is basically to identify your DNA source and isolate it. So I'm very low tech in my lab. We use yeast. So these pictures were all taken in my lab. Yeast is our model organism. You can't see the mess on the bench, but it's a very, very messy, non-clean place. So we isolated our DNA from the yeast. And this is a multi-step protocol. Lots of mixing that would have to be done on a chip and vulnerable to attack in those steps. I'm going to talk a lot about, so PCR is the assay that we decided we were going to use simply because it's ubiquitous. So most protocols that are being miniaturized onto chips use some aspects of PCR, so that's what we decided to use. And we're also, we'll get to this a little later, we can visualize that with the naked eye by running gels. So just to walk you through what is PCR, if it's been a while, it's polymerase chain reaction. It's something that was discovered in the early 80s. It was a really basic biology process of how DNA is made. And then captured by industry for different uses, sort of, and basically doing some cloning aspects, and then basically using for analysis and all these different applications. So the real key that you need to understand about PCR is that you start with a very simple low copy sample source, and then you have something that you're looking at. So maybe you're looking at a polymorphism, you're looking for some particular sequence in the genome, and so you have some target. And how you find that target is by having specific primers that you engineer to find that particular part. So by using those primers, you can start with one piece of DNA, go through the process, which gives you 2 strands, go through the cycle again, give you 4 copies, on and on and on, until you have millions and millions of copies of what you're looking at your target sequence. And that's how it's done. Okay, so what I took for granted and what Mohammed was interested in is that simple process that everybody uses every day requires a lot of steps, a lot of solutions, and is probably very vulnerable to attack. Okay, so that was sort of the bottom line. So once you get these solutions ready to go, and once you're ready to do the experiment, then there's other things where we are going to prepare the samples and particular controls. So that comes into assay design. So good assay design has a positive control and a negative control, and then all of our DNA samples that we isolated, I think we're going to walk through, right? So this is just a basic benchtop science in experimental design, which is, I'm sure, incorporated into all of your assays. As we heard about at babies, they have their biologists go and sit down at the computer and say, I want to mix this and this. So they have that all built in. But at the benchtop, we were doing it on a macro scale just to see what could go wrong. Okay, we used an old-time PCR machine basically to do this. So all of those small mixtures are put into a thermocycler, goes through many, many cycles to copy that DNA over and over again. Okay, and then we wanted to set this up so that it was something that we could see visually. So in order to see the products of this reaction and really model what an attack looks like, we used, again, sort of an old school type of analysis, which is agarose gel electrophoresis, where we take the products of that PCR reaction, put it on an agarose gel, run that based on the charge of the DNA. And so very simply, what we're looking for at the end of this reaction is some kind of pattern. So we have a control, which is going to have a particular pattern, which is going to show up as bands on the gel. And then our experimentals are going to have a different pattern, maybe different size, different number of bands. But that's the pattern, and the presence or the absence of bands is what we're looking for. And that's going to be our readout analysis. Okay, readout and analysis. All right, so the first attack that we modeled was a positive denial of service, okay? And we decided that the best way to prepare this was to have the attack happen at the level of preparing the PCR mix. And so how did we implement that? We deliberately added a small quantity of our positive control to every sample, okay? And then went through the steps of the PCR analysis. Okay. The second attack that we modeled was a negative denial of service. And again, we targeted the PCR master mix for that step. And the way that we implemented that was to add a chemical to the master mix, which went to every single experiment. And then attack number 3 is a forgery, a DNA sample switch. So we modeled this attack to happen at the point where we were preparing our samples. And implemented that, again, just by literally closing our eyes and moving samples around on the bench and then going forward with the rest of experiments. So very low tech, but the principles are what's going to drive our understanding of how stealthy these attacks could possibly be, what that looks like, and potentially how you can prevent that from happening. Okay? So we essentially set up a control assay where there was no attack, and then in parallel, attack 1, attack 2, and attack 3, and then we ran all of those on the gel to look for, to detect the results. And so, go ahead.
Yeah, sorry, I had a question. Where did the supply chain will the attacker sit for the and deliberately put these samples in the...
I'm sorry, I missed the first part of your question.
In the supply chain.
Where in the supply chain? It has to happen either in the DNA collection or in the preparation once it gets to the lab or wherever the assay, wherever the microchip is going to be run. I can't, where, our journalist, did he leave? Because I'm going to show some really ugly data which has not been manipulated. Okay, anyway, so this is what we're going to be looking at. So this is the, so as I said, as you go through PCR, you amplify and amplify and amplify.
Oh, Charles.
Yeah, and then in order to see it, we run it on a gel where it intercalates with some UV chemicals that allow us to see that on a UV gel. So we're really going to be looking at these bands on a gel. This is sort of idea. Right.
Oh, this is your Western blots moving up.
So Western blot is proteins, but this is a DNA gel, so we're just looking at DNA here. But it's exactly the same. So very, yeah, we could manipulate these images if we really wanted to. And as you'll see, they aren't beautiful. We did not manipulate anything. Okay, so we're going to interpret I'm going to interpret this data for you. Okay, so we're talking about number one. This is how the experiment is supposed to work. So in lane number one is our DNA ladder. So those are just the known sizes. We know what the size of that product is, which allows us to say, the gel looks good, and it gives, this is really part of what I'm talking about, but we can measure what the size of that product is. Okay. In well number two is our negative control. So if it's a negative control, you shouldn't see a band. We don't see a band, which is good. In the next lane is our positive control. We expect to see a band in positive control. In lane number three, we see a nice band. It's the right size, so that's great. And then the rest are our samples that we scraped up off of the yeast plate, and then basically asking, what does that target look like in each of these independent samples? And so in lane four, yes, we have a band. It's the same size as the positive control. In lane 6, there's some kind of DNA deletion there, so there's a polymorphism. 6 and 7, there's no bands. We know the assay worked because our positive and our negative controls worked. So that's giving us a negative result for these two particular samples. And then 8, we have a band, which is a particular size, and 9, we see that deletion as well. So this is the control experiment. This is what we expect to see. This is how we would expect to see it look. Okay, so in portion 2, which I'm going to uncover in a second, was attack number 1, which was the positive denial of service, right? Okay, so we manipulated, we attacked our master mix with a positive control. So what are we going to see? Bands in every gel, bands in every single lane, right? So this is, something went wrong, and we can tell several different ways. Our negative control has a band in it. That's our first indication that there's been an attack. And that's really the only indication, because if we had not done this first experiment, maybe that was the real answer. We just don't know. But because of the experimental design, because the negative control actually has a band in it, we know that that experiment didn't work, or that there's been an attack.
Or could you manipulate the negative control and not touch that?
Well, that's, yeah, right. So what we would do in real life is we would say, yeah, either there's been an attack or I screwed up at the bench, so let's repeat it and see what happens. And reproducibility kind of falls into this as well. So here's the negative denial of service, right? Nothing, absolutely nothing is shown. So again, that's easy to interpret. You don't know where it went wrong, but at least you can see it. And then attack number 3 was the sample switch. Now this one's a little more interesting because it looks just like 1 in the ones that we know about. In our negative control, there's nothing. In our positive control, there's something. And then in our experimentals, some of them have bands, some of them don't. They're different sizes. But this could be the result that I give back to the crime lab, and I say, it was suspect number 4. I don't know that there's been a malicious attack. Okay, and so this is the conclusion sort of modeling these different assays, is that we can model them, and what we learned from this is that the sample forgery is the most stealthy. which is not, a large conceptual jump to take. But now we actually saw it. We ran through the steps. And the question is, okay, what can we do to prevent that? What can we do to stop that? Is there anything we can do? And as I was listening to the talks this morning, I guess one thing I was thinking about, where you were talking about reusing chips and counterfeiting chips, I guess that would look like this, right? So if somebody was counterfeiting a chip or reusing it, it would look like a negative denial of service. Okay, but so what do you do about this attack at the level of the sample? So I'm going to propose a biochemical level countermeasure to prevent stealthy attacks. Okay, so this is the theory of how we're going to do it, and I'll just give you the upshoot. It's a lock and key type counter mechanism. So here's the idea. So when the sample comes into the lab, it's given a particular barcode. And this isn't a visual barcode or an electronic barcode. It's a DNA barcode, a biochemical barcode. And that gets added as early on in the process as collection. Okay. And so that's really the key. And then the analyzer is going to do the experiment, and they're going to do a check. Does the barcode match what it should be, or does it not? And that's going to inform whether they go through with the experiment or they stop. So we're incorporating an early validation step in our assay. Okay. So what is our barcode? Our barcode is actually DNA. So again, part of the design of this experiment was to do something that we could see. So we designed our barcodes to be a kind of large, 280 base pairs. But the idea is that every sample would get a new fragment of DNA added to it independently, not integrated into that DNA, but added separately of a particular length. And then depending upon that length, that tells you how many different barcodes you can generate. So if you wanted to do 100, if you're going to be running things like on a 96 well plate or a 396 plate, you would basically want to do barcodes that are maybe 7 or 8 base pairs, and then you would get a whole bunch of unique ones, one added to each sample when it comes into the lab, and that would allow you to go ahead and do that experiment. But that's the theory behind it. Okay, so the barcode is the lock, and then the primers are going to be the key. Okay, if you know what the barcode is, then you can engineer or design the particular primers that are going to amplify that barcode. So that's the second step. And the key, as we write here, the key here are barcode-specific primers. Okay, so the question is, does this work? Okay, well, so this is, again, this is a little bit more just to reiterate what I just said. So the idea is the sample would come into the lab, it would get a secret ID, and it would get, recorded somewhere safe under encryption that it was added to it was barcode 1, and that we know what barcode the particular primers are. So that would go into a secure database. The analyzer, the collector would only get barcodes, and the analyzer only gets the primers or the keys, right? And so as this goes through, we unlock the key, and we see that when we do that, we get a positive result that tells me that my sample is correct, and I can continue on with the analysis, or the sample is not correct because the key did not unlock the lock, and we have to stop the analysis because we know there's been an attack. Does that make sense?
So I think at this point, I'd like to add, we have 2 channels for communication. One is the biology channel, and the other is the electronic channel. And the 2 are being done at the same time. That's how they add each other.
Thank you. Okay. So does it work? So this is where a lot of the optimization comes in, and sort of optimizing it really for what we wanted to do. So when Mohammed and I when Mohammed came into the lab, we wanted to go simple, and we designed 6 different barcodes with 6 different primer sets. And I'm only going to show you 2 here, and I'm only showing you 2 because these are the only 2 that worked the way we wanted them to work. So the first step was really optimizing, so designing the barcode and designing the primers.
Like example of falsification.
Yeah, I'm being 100% honest here. There is no, I'm not not hiding anything. So designing the barcode and designing the primers to amplify that piece of DNA was easy. What becomes difficult is thinking about when you're adding these to a biological sample isolated from nature or from the lab, is it becomes a question of quantity and ratios and things like that. So we had to go through and really showing you the end result of some work here. So what we really wanted to do, so question number one was, we have to figure out what the right amount of barcode is to add to each sample. You can't just add it crazy, because that's going to interfere with the rest of the process. So this is sort of the first step of that, where we took barcode only, no genomics, no DNA from the yeast or the sample, and did a serial dilution and asked, what is the least amount of barcode where we can still get a signal? And these, as you can see, these are show you two really different results that we got. So at the same dilution, 1 to 1,000, we get a really robust over here for number 2, because you see you can go many more dilutions on this barcode. And then this one just drops off as you go from 1 to 1,000 to 1 to 10,000. So there's a lot of optimization that has to go into that step of it, but we got that working and we were happy to do that. So we moved on. So the next step was to take those barcodes and add them to a genomic sample and then question it. So then we were going to do multiplexing. So to me, multiplexing is we're looking for 2 different products in one DNA sample. Okay. Right, so that just explains that. And this is sort of the theory. This is what we expect to happen in this multiplex. In the tube, we're putting the sample and those target primers. The barcode has already been added when the sample was collected, and now the analyzer is analyzing those is adding those barcode primers. And then the expectation when we run that on a gel is that we're going to see 2 bands, one corresponding to a known size, which is the barcode in blue, and then the sample band, which is the unknown. Okay. And so for these 2, it did work exactly as we expected. I'm not sure about the resolution here, but for sample 1 and barcode 1, there you can see there's band number 1 and there's band number 2. And again, we see that this is very sensitive to dilution, because once you get a little bit more dilute, you're losing your sample band here. So it only worked in these specific. Oh. Yeah? Yeah, so it's very, very sensitive to dilution. And then in the second example, we were able to take it out to a little bit more dilute. But again, we saw band number 1 and band number 2. So for both of these barcodes, we were able to optimize them and get them to work with our genomic sample that we isolated from the lab. So there was one more step that we wanted to do, and that's outlined here. So this is a cross check. This is looking at it a little bit differently, but so in column 1 here is what I just showed you when we do sample 1 plus the barcodes. Column 2 would be adding in those primers, which are specific to the barcode. And then in the third lane, we wanted to know, okay, what happens if we add in a different set of primers? So let's say that I'm the analyzer, and I was told to add in barcode number 2 primers, but I make a mistake and I add in barcode primers number 1 anyway. So how rigorous is our assay in this way? And there's many other manipulations we could do. This was just sort of the start of where we wanted to go with it. And so these are sort of what I refer to as our ugly data. So we had some different results, which are a little bit inconclusive at this point. So in region 1, things worked as we expected them to. So here we sample only, so we saw the 2 genomic samples. When we added in the barcode-specific primers, in the green, this worked as expected. So here's the original sample band, here's the barcode band. But when we moved in and added more primers just to see if it would amplify something different, we kind of lost that specific band here for the genomic sample. And then in sample number 2, in sample number 1, we saw the genomic band first, but then we lost it once we added in the barcode primers, and then really got some uninterpretable results once we added in the extra set of primers. So feasibility, it seems feasible, but there's still some work to do in order to get this to be a really robust assay. So more optimization is needed. And again, we started with 6 very, very simple barcodes. We worked with 2 of them, so we clearly could scale this and do some work in the lab to figure out something which will work. Yeah.
On this particular point, do you think this is more of a protocol issue or more of a design issue with the primers?
Yeah, that's a good question. So the primers work independently, so we can amplify barcode by itself, we can amplify sample by itself. It's when you mix things together where you're not quite sure. So we're adding in barcode primers, which are designed to be specific to the barcode, but they're able to sit down anywhere in that genome. Which even for the yeast genome is 15 million base pairs, which is very tiny relative to a human genome. So what we think is happening is that those barcode primers, even though they were designed to be optimized to the barcode itself, they're finding other places to sit down in the genome. And that's why we're getting some anomalous results once we do the mixing. So independently, everything works as expected. It's when we mix it. So theoretically, either trial and error or intelligent design, primer barcodes and barcode primers, which are specific only to that barcode but not to the genome, should be identifiable. It's just we weren't lucky on the first try. I don't think it's protocol because it's PCR, very simple protocol. Anything else? Anyone else? Yeah.
It seems, this could be taken beyond. Some work I did like 10 years ago, we were looking at whether or not, the existence of a DNA database, where basically it's authentication. Basically, you want to say you basically have a regiment, a military regiment, and you have one cocktail that has, some portion of their DNA in the cocktail from all the members of that regiment. And now you come in and you basically you can aliquot that to a whole bunch, and then you find someone on the field, you take a single sample and add it to it and say, and you're just asking, are they in that regiment or are they not?
Right.
And so, and so it's in that time we were using isothermal RCA rolling for quantification, because then you could the thought was, in a remote field, we could just do a mix and read, I got a color change, that person's in the biological database.
Right.
It seems sort of similar to
Exactly. So yeah, it's some kind of biological something that you add to these samples very early on in the process, which really then allows you to sort of do lock and key and say, yes, this is right, no, this is wrong, very, very early on in the process, right? And so I agree, this doesn't have to Yeah, I think the idea is it would have to be something that's added to the sample at the collection site or very early on. So, and it would have to be something that was individualized to that sample. So, some type of unique fluorescence, maybe like a fluorescence. It would probably still have to have some DNA sequence specificity to it, because that's where our specificity is coming from, is being able to bind to the DNA. So the primers really for the PCR amplification is where that's coming. But your sample coming into the lab, you have no idea what that is, right? So the lock has to be independent and work by itself, independent of that sample. So fluorescence, maybe binding to I think there are more possibilities. We just haven't really thought through. What are you thinking of?
Yeah, sometimes use like this, particles.
Particle.
Coated with some, pattern, and then you can this pattern and just microparticles with specific pattern into this.
Right. So a little bit like IBM was talking. I forgot your name. I'm sorry. I'm talking about earlier this morning, right, where he had the color signals that was going to be unique to each and every biochip, right. And then you would detect that early. Right, so these are just our conclusions. We demonstrated preliminary feasibility for a counterattack to these stealthy forgery attacks. However, experimentally, this is a bit of a heavy lift. We have to do a lot of optimization and able to do it the way that I've described it. As I mentioned earlier, we used fragment size as a distinguishing feature for our benchtop demonstration for visualization purposes, because we wanted to really be able to see it on the gel, but that could be changed to many different things, or even smaller DNA is probably going to be much easier to work with rather than